all to the day first of the Nobel Evening Talk Series 2021. First of all, we are thankful to all who signed up for this event. If you have any questions, suggestions, or comments, feel free to put them in the chat box we will take at the end of the talks. Now, I would like to introduce our first speaker of the day, Dr. Nixon Abraham. He is a neuroscientist and assistant professor in the Department of Biology at IISER Pune. He obtained his PhD from Max Planck Institute for Medical Research and postdoctoral from the University of Heidelberg. Throughout his research career, he has been awarded with various fellowships and prestigious awards. With these few words, I would like to invite Professor Nixon to talk about Nobel Prize in Physiology or Medicine. We welcome you, sir.
Okay, as I said, if you don't mind, I will switch off my video as I'm presenting from home. I hope that's fine.
Yes,
Yeah, yeah. Okay, good evening, everyone. And thank you very much for the introduction and for giving me an opportunity to present the novel work. Although this is not my domain, but I will try to summarize in simple words what they have done to get the Nobel Prize this year. Okay, let me share my screen. Can you see my screen?
yes, sir. Screen is visible.
All right. Okay, so I put a title called Spicy Touch. Spicy Touch. So this is a cookbook written by some authors, and you will get to see why I use this title. Okay, when we get into the details. So here is the announcement. So the Nobel Prize in Physiology and Medicine was awarded to Dr. David Julius and Dr. Adam Potaputia for their discoveries of receptors for temperature and touch. So that is for revealing the detection mechanisms for temperature and touch stimuli. How do we detect these stimuli? Before starting with the details of the novel work, let me explain some basics. So the question is, how do we perceive the external world? Of course, we use our sensory organ, right? And all of you know that we have 5 sensory organs. And our sensory organs collect various stimuli present in the surroundings, and then they are processing this information and doing something. Lots of processing is happening, and then this information is going to specialized relay stations before it reaches the cortical areas. And these are common things across different sensory systems. Okay, and by now we have really good knowledge about how the information encoded in these specialized relay stations, as well as up to some extent in the cortical areas. And I'm sure that you guys have seen this sort of colored pictures from the different websites or textbooks. And what it says, there we have in the cortex, we have specialized area that controls the information encoding through various sense organs. And if you look at the history of Nobel Prizes, so there were prizes awarded before the information processing, for the information processing through different sensory systems. For example, in 1961, George von Bexy was awarded with the Nobel Prize for his discoveries related to the mechanisms of stimulation within the cochlea. And then the Nobel Prize in Physiology or Medicine in 1967 was given to these three guys, Granite and Hartline and George Wald, for their discoveries related to the visual information processing in the eye. Then our favorite topic, so the in olfaction, the Nobel Prize was given in 2004 for the discovery of the odor receptors and the organization of the olfactory system. And then this year, it was awarded to Dr. David Julius and Dr. Adam for the discoveries of receptors for temperature and touch. Okay, so whenever you think of the information processing starting from the external world and then reaching our central nervous system and then forming a percept. So there are different steps involved in it. And so two major steps. One is sensation and the other one is perception. So how do we define these two important steps? So actually these are two complementary stages of information processing. And what happens by completing these processes, a neural representation of the external stimuli is getting formed in our brains. So what happens in sensation? Through our eyes, it's light, and through our ears, it is sound, and through our nose, it's smell, and tongue taste. And what happens actually in the sensation process? So these physical or chemical stimuli are getting converted to neural signals. Okay, and the perception is the process in which these neural signals are then sort of defined or interpreted by our higher brain centers. So these two processes are absolutely needed to perceive the external world, and depending on the perception that we form, I mean many stimuli. If you think of any sensory stimuli, right? For example, if you get something related to, if you see something related to danger, you just run away. Or if you are touching a hot plate and then you withdraw your hand, right? So in many occasions, the percept formation is absolutely necessary to to do some actions. And in some cases, we have to do the actions even before a complete percept formation is happened. So there are some funny examples that I always tell in my class. So I did my PhD in one of the max banks in Heidelberg, and one day there was a fire alarm, and then all of us we got outside the institute, and there the fire force came very fast, and they got into the building, and then finally what they found was that was a burnt pizza that caused all these things. So in certain occasions, you don't get time to react, or you don't get enough time for the complete percept formation. So that can also happen. Anyway, so the goal of this process is responding to the ever-changing environment. So to show the complexity of this process, how complex the perception, right? Let me just show you my favorite example. So the Leaning Tower of Pisa, and I'm sure that all of you are seeing that this the right one is more leaned towards the right side, right? And I don't think that any of you have a different opinion. And if you look at this, both these pictures very carefully, so this is the first one, and the second one. Okay, so it's the same, right? There is no difference. So, but what happens here, so our visual system is treating these two images as a part of one single scene. So that's why this kind of illusion happens. And what I wanted to convey, the physical dimensions are not equal to perceptual dimensions. So it's a very complex process. Anyway, now what happens at the sensory periphery, right? So if we look at our skin, if we take that as an example, and it's a very interesting organ because it can process many different types of stimuli, right? So the nerve endings can be sheathed, like for example here, or unsheathed like this. And here is the thermoreceptor that senses the heat or cold. There is the mucinous corpuscle sensing the touch, and the nociceptors for sensing the pain, and then Pacinian corpuscle for sensing the pressure, so on and so forth, right? And so it's a very interesting organ just because it can sense various types of sensory stimuli. So let's just focus on the pathway of the pain perception. Okay. So you take the example of any noxious stimulus, right? And what happens? So there are different phases. So one is the transduction. So that happens here. So at the sensory periphery. So here, the nociceptors convert these noxious stimuli into electrical signals. So then these nerve fibers are carrying these electrical signals to the spinal cord, and the cell bodies of these nerve fibers are lying in dorsal root ganglion, and from there the dorsal root ganglion is lying just beside the spinal cord, and there the second order in the spinal cord, the second order neuron is taking the signals further to the thalamus and to the cortical areas, and then our central nervous system perceive what we have sensed. And this is how the perception of a nociception happens. And then there are also the descending modulatory fibers happening, and there are a lot of modulations, excitatory as well as inhibitory, that happens in the spinal cord. And as a result of all this, finally these signals are getting relayed to the higher centers, and then we form the perception. So this is how the pain pathway works in our body. Okay, now what happens at the sensory periphery, and how are these stimuli detected? So this is the question that they try to address, and I'm going to talk now about David Julius's lab work, and he was one of the recipients, and this is the major paper, and there are also many other papers which are important, and you can find those papers if you look at the Nobel Prize website. As I said, I'm not, as you guys are coming from different backgrounds, I'm not going to get into the details, but if you have questions, please ask me anytime. Okay, and so what Julius Lab was, what they were interested in, using natural products, for example, the toxins from tarantulas or capsaicin from chili peppers, and the pungent chemicals from horseradish or wasabi, this kind of natural chemicals, and how they are modulating the pain or the temperature perception. This is what their broad interest was, and they started to work with this capsaicin molecule, which was the most interesting candidate at that time. So a little bit of history. So in 1970s, the Jansko and colleagues from Hungary, they showed that the capsaicin can elicit a sensation of burning pain. And few years later, these guys, the Bevan, Rank and colleagues from London, So they studied the biophysical properties of sensory fibers, and they looked at how the capsaicin is interacting with the sensory fibers. And what they found was the capsaicin can depolarize the membrane of these sensory fibers, and they can also cause the sodium and calcium influx. Okay, now the question is, capsaicin, it's a vanillin compound. Is there a specific receptor for it? Okay. And it was thought that there is no specific receptors. Somehow, this is interacting with the lipid membrane. Okay, so this was the idea that was there in the field. So they wanted to really go for finding out the receptors for the capsaicin. So their approach was very simple, and so as nothing is known about the receptors, it's completely blank, right? When they started the experiments, so they went for a structure blind and identity blind approach. So their idea was taking this messenger RNA pools from the dorsal root ganglia, and I showed you here. So here, this is the dorsal root ganglion, right, where the soma of these nerve fibers are present, right? So if you take this one, so whatever the RNAs are present, and their receptors are at the skin surface, right, and which are receiving these different types of stimuli, and as these receptors are getting activated somehow, and so they didn't know at that time the receptors, right, but there will be some sensing mechanism, and this some sensing mechanism will be active, and therefore what, because of the activation, whatever the changes that is going to happen, it can be picked up if you isolate the dorsal root ganglion neurons, and if you look at the mRNA levels. Okay, so what they did, they decided to take out the mRNA pools from dorsal root ganglia, and then importantly introduce this to non-neuronal cells that are insensitive to capsaicin. So there was a reason, and then expose to capsaicin. Okay. Now the question is, so if their hypothesis is going to work out, if they are doing this, suppose, so they could take out the pools of mRNA, and then mRNA is not very stable. So they generated the complementary DNA libraries from this one, and they had many pools, and they were going to put this in the cells in the absence of capsaicin and in presence of capsaicin. So when they did that, say they looked at the calcium activity that happens in different pools. Okay, here for example, there is no fluorescence present here, right? And in this pool, there are few cells that shows the calcium activity. Why the calcium activity is important? It is important because calcium is an important candidate that controls the neural activity in our brain, right? So for the non-neuroscience people, so how the electrical impulse generation happens, right? So you have the neurons are connected. So the neurons are connected from one to the second one, from the second one to the third one, so on and so forth, right? And when you have these neurons connected, so what happens? One neuron is, for example, the sensory neurons that we just discussed. So the sensory neuron is getting activated, and then because of the activation, the sensory neuron, the electrical impulse generation happens in the sensory neuron, and then this electrical impulse travels down the axons and it reaches the nerve terminals. And at the nerve terminals, there are voltage-gated calcium channels, and these calcium channels would then allow the calcium ions to get inside. And this calcium influx is very important for the neurotransmitter, for the chemical release from that specific neuron. And then these chemicals are now going to the second order neuron and binding to the receptors. And the same process continues, right? The electrical impulse generation. And so they wanted to look at the calcium, the activity of the calcium indicators, and they decided to use this FURA tool, which was developed by Roger Chen. And all of you might have heard that. So these three guys, Shimomura and Shalfi and Roger Chen, got the Nobel Prize in 2008 for the discovery of the GFP. Okay, and here in this pool, they could see certain cells with the calcium activity, and then they further separated this pool, right? And finally, until they got a single cDNA that was encoding for the capsaicin receptor, and then they named this as vanillin receptor 1. Okay, now as the next step, so what can be done? So now you have the cells that are showing this kind of calcium activity, meaning that there is something happening in presence of capsaicin, right? And so potentially there could be some receptors present in this one, and they identify the receptor. And now if you want to study how the receptor functions, and then you have to really go for the electrical activity, for looking at the electrical details of the electrical activity in these cells, right? So maybe let me switch on the video for a few minutes, because maybe it's good to explain this thing. Okay, so they got the Nobel Prize for measuring the currents from single ion channels, and then they really discovered this technique called patch clamp method. I don't know. You can see my screen as well as me, right? So what actually happens? So how do you do this, right? So you have these membranes. It can be the membranes of the neuron or the membranes of the cells. Okay. So you use a glass pipette, and then you generate a positive pressure in the glass pipette, and then you approach the membrane, and then if the membrane is like this, you're pushing the membrane with the glass electrode, and as the positive pressure is there, and this glass pipette, there is a metallic electrode which is then connected to the amplifier, and to amplify the signals that they are going to receive from this membrane, right? And then they push the membrane a little bit with the glass electrode, and then with the positive pressure, and then they release this positive pressure, and then what happens? So suddenly there is a seal formed between the pipette tip and the membrane. Okay, and so that seal should be of giga ohms, the range of the giga ohms. If you look at, there is no lead current at all. If you manage to get the seal like that, and then now you can think of doing the recordings in different modes. For example, you can keep the configuration like this, with the cell attached mode or the membrane attached mode. And now, as there is no lead current, whatever the changes that happens here inside, that can be picked up to the metallic electrode present in the inside the electrode. And so that should be coming from the ion channels present on the membrane, right? So then if you want to go for single ion channel studies, you can even, these experiments are not very easy. You can take out the membrane, you can pull the membrane, and then you can get a piece of membrane attached to the glass electrode. And then you can keep either like this, the inside out, or outside the outside out, like that. If you allow these two ends to anneal, and then this outside is coming exposed here, and here the inside is exposed here. The idea here is, if you want to go for the neuropharmacological experiments with this kind of different configurations, you will be able to modulate or modify the activity, the electrical activity that happens inside as well as the outside of the membrane. And these guys discovered this technique, and I did my PhD in this department. And one of his famous quotes is "Einmal is keinmal," so meaning that one time is nothing. So you have to always reproduce. If you go to him with some experience after finishing one experiment, then he always asks you to go back and then repeat the experience. Einmal is keinmal. Anyway, it was fun doing PhD in this department. So now coming back to the presentation. So what they did, right? So they did nothing but, so they took. I told you that they had this fluorescence from the cells, and and they could finally identify the sequence of the VR1, Vanderbilt receptor 1, right? And then they decided to test many agonists and antagonists. Okay, and then they also express this VR1 gene in synapse suicide. So that is another famous model. If you want to study, the electrophysiological techniques, you can express, and then you can use it. It's a big cell, and because of the size of the cell, this kind of experiments are relatively easier with the synapse suicide. And in presence of when they did this, when they expressed the VR1, and then when they put the capsules in, what they could see an inward current. Okay. Now the question is whether this what can modulate this inward current. So there is clearly the ion flow happens because of what has been expressed, right? And then they looked at the activity of many different agonists as well as the antagonists. For example, here uh, capsaicin, that's an antagonist, and butylenide, that's another antagonist, and they could by applying this antagonist they could get rid of whatever the inward currents they were getting in presence of capsaicin. And if you wash out these antagonists again, you can see the inward currents coming back. And they could even see the differences in the responses. Here is the percentage capsaicin response, and here is the concentration that is plotted. And when they varied the concentration of the capsaicin, they could even see the differences in the activity, the extent of activity. Okay, now the question is, so you have this receptor expressed on the cells, and what are the ligands? Okay, and they were trying many different substances to find out what can actually activate this, right? And so this is his own wordings, and the idea of heat came through the experimentation, where we just kept asking what causes pain and what kind of things we can throw at these cells that are expressing the receptor. Eventually, we tried heat, and that heat really worked out. And when they, so these are again the non-responsive pools, and then here is a VR1, and then when this the cells that were expressing this VR1 receptor, when the heat was applied in the extracellular solution, and they could really see again the calcium activity in the cells. So what does that say, right? So the heat is really activating this ion channel that is present on the membrane. So that's the takeaway message from these experiments. So now capsaicin can activate and heat can also activate. And so, so later it turned out that the sequence for the VR1 receptor is the same as the transient receptor potential channel that was reported. This receptor was reported in the Drosophila, and where because of some mutation in this receptor, they could see the defective vision, but nobody knew until this work came what it does, right? So they named it as a TRP V1 receptor. And then if you look at it's a wide family of genes. And later, Adam, the second recipient of the Nobel Prize, and he also independently found another variant of this TRP, which is named as TRP M8, and which is sensitive to the cold temperature and menthol. And so this TRP M8 receptor allow the calcium influx under cold conditions. And now, so here is the summary of the entire study. Um, you can vary the heat, the temperature, and you can, when it is going to the colder temperature, then it becomes noxious, and then this TRP M8 channel is opening, and which can also activated by menthol. And here, when it goes to the higher temperature, this TRP V1 channel is getting open, and which is also activated by the chili, the capsaicin present in the chili pepper. So now, why I named the talk the spicy touch, right? The spicy part is now over, and then later in this paper, they reported the knockout mouse line for this TRP M8 receptor. And what you see here is, so if this channel is present, they have this chamber, and you can see the video of this experiment on YouTube. And there are two chambers, and this part is heated up to 30 degrees, and this part is kept at 20 degrees centigrade. And if you allow the animal to explore, the animal goes, a wild type animal goes, and because as it can sense this temperature here, it doesn't go here, it just stays back. It's a very simple experiment. And then if you allow this knockout animals to explore, because it cannot sense this temperature difference, it can walk, explore everywhere. And so that was very clear from the video. I just took a snapshot of that video. Anyway, so this study is very elegant because he went all the way from identifying this TRP V1 or TRP M8 molecule from the molecule level to the behavior level, right? So that's something that is very extremely challenging in neuroscience. So going from the molecule level and then studying at different levels and then dissecting out the behavioral relevance, it's very challenging, and therefore this is an elegant study, and that's why they got the Nobel Prize. Okay, so because of the limitations with the time, so I'm not going to get into the details, but the structure is also known now. The structure of TRP V1, and this was a news and views published by Henderson, and the structure was published in this paper, published in Nature, and basically they have identified the structure using cryo-electron microscopy, and then you can now we know the binding site for example, the spider toxin. So these toxins can also modulate these channel functions, and then capsaicin also, and some other toxins which are the analogs of capsaicin. So this arrow indicates the calcium influx. Anyway, the structure is known, and if you have any questions related to this and or related to electron cryo microscopy, I think you should go to Sai Krishnan. Okay, now coming to the touch part. So do you have any questions up to now, or can I continue, or can I ask? Can we discuss the question at the very end? Do you have any questions? Can someone answer, please? No? Should I continue? Okay, so the touch sensation. So if you think of the touch sensation, can you guys hear me? Am I audible?
Yes, sir, you are audible.
Okay. So if you think about the touch sensation, there can be different types of touch. For example, active touch or the passive touch. And I'm not sure whether you identified this person, Nicolas Sarkozy and Carla Bruna. And then there can also be emotional touches, and if you think of the other functions, right, where these touch receptors can be involved, the proprioception, for example. What is meant by proprioception? So we have and perception uh, on the three-dimensionality of our body parts, right? Even if we sit closing our eyes, uh, we can see that we realize that where is our legs, where is our hands, and all these things because of the action of these touch receptors, and it can have various functions, and so, for example, controlling the blood pressure. And once our bladder is full, how do we know that? And these receptors are also important in sensing that. And now the question is, how did Adam's lab find out these candidates? Right. Let's see the details. So, if you think about their strategy, so let me just repeat what David Julius did. So David Julius took the cell line where they were, it was a completely blind approach, right? And and those cell lines were not really sensitive, right? Therefore, they had to really go for, and by pulling the cells from the DRG and then going blindly and then finally putting the capsaicin and then looking at the responses. Here they did an opposite. They did an opposite thing. So they wanted to start with some cell lines which are sensitive to the mechanical stimuli. So they have screened several mouse and rat cell lines, and what they did. So of course, how do you do this? So the same patch clamp technique that I explained, right? So you have to really stimulate the cells using, and here they used the piezoelectrically driven glass probes and to push the membrane, and at the same time, so here is the mechanical impulses given by this piezoelectrode, and then at the same time you have to record from the cells, right? And when they did that, so they could see the currents happening because of the mechanical sensation that happens in this cell line. Okay, and after screening through many different cell lines, so they finally they could see these currents happening in this neuro 2A cell line, which is nothing but mouse neuroblastoma cell line, and they shown the most consistent mechanically activated currents. And then so they could see the currents. And now what happens if you do the opposite thing? Okay, instead of putting the pressure, you stretch the membrane, so you can apply the negative pressure. So this is exactly what they did here, and even in that case they could see the currents unfold corresponding to this mechanical stimulation. And then what they did, so they wanted to, now the question is, so they had a cell line which was responding to this mechanical stimuli. Okay, and what causes these responses, we don't know. They didn't know at that time. Okay, so they obviously you wanted to abolish this responsiveness, right? And then they decided to go for this the technique called RNA interference technique. And how did they do this? Okay, this technique also got so for the Nobel Prize. So that is Fire and Mello in 2008. So now, so they have to really test what happens. So they have to first find out the candidate molecules that they have to knock out, and again they screened and they tested around 80 proteins using RNA interference. And what the RNA interference technique does, it uses a complementary messenger RNA to abolish an existing mRNA, which would otherwise be translated to a protein. Okay, like this, you can shut down the activity, or you can knock out. And they had many candidate genes screened, and then they decided to use these screened and selected genes, these 80 genes, And the idea was out of this 80, when they use one of these proteins and using this RNA interference technique, they will be able to identify the sequence. And so what you see here is using this siRNA technique. So you always have to have this control of this putting the scrambled RNA fragments, and then using this targeted RNA, you can abolish whatever the currents they were visualizing here. And out of this 80 candidate proteins they screened, they could select one sequence that was encoding for the protein, and then they named it as PSO1, and this was this means that the Greek word means it pressure, and this was the sequence that was encoding for PSO1, and this was the sequence that was encoding for PSO2. Okay, and therefore they could by shutting down the activity using the RNA interference method, now they could find out the sequence of this specific candidate molecule. And once you have the sequence, what you do, then you can again go for any other non-neuronal cell types and then express the sequence. And then you do exactly the same. That's what they did here. And they express this in HEK293 cells. These are human embryonic kidney cells. And then they expressed it, and then the mechanical stimulation was done, and they could see the corresponding ion flow or the current flow through these in these cells, recorded from these cells. Okay, and now, so these piezo channels are present in many different tissue types and in different animals, and they are also present in plants, because the sensing the pressure and the stretch is something very important. Okay, if you think about different functions, and so they are also involved in various functions, and they have important medical applications. For example, and I happened to see one of the interview, and he was asked the question, can Adam tell about a potential medical application? And this is what he was telling, and I'm sure that he has the data by now for making that statement. He was saying that this PSO2, so this can be used to treat neuropathic pain, and then he explained this condition, this tactile allodynia, and it's nothing but you feel this innocuous touch as very painful. And what he was saying, blocking these PSO2 receptors could help the patients. And so it has various applications also. Now, before I conclude, let me tell you why we are excited about these findings. And the findings are there, but as Nobel Prize now was given to these findings, I think it's a big positive thing for us. The reason is this. So we work on the olfactory system, and in one of the ongoing projects in the lab, what you see here is an behavioral setup that we developed, and the animal is sensing the airflows that comes from these two locations here, and here the airflow is coming in a diffuse manner, and this airflow is without any odor molecules. Okay, so whenever we train the animals like this, what we observe was the animals were using their nose to sense these airflows. Then when we train these animals to discriminate various airflows, for example, 8 liter versus 4 liter, and up to 300 ml versus 800 ml. Okay, and in all these cases, the animals were able to differentiate these different airflow rates. Okay, and when we trained the animals for the same airflow rate, there was no learning involved. So this experimental results show that the animals can detect and discriminate various airflows. Now what we did, we took out the olfactory sensory neurons in the nose, and then we took out also the olfactory bulb. Then these animals were not able to differentiate these various airflow rates. So like that, we can now clearly say that the rodent olfactory system can act as an animal detector or discriminated. Now the big question here is how do they do this, right? Why this is functionally relevant, and this is going to be the topic of our industry for the next few years. And we have some indications, and we looked at the expression of these PSO2 receptors in the olfactory bulb, so which is very specific to the olfactory information processing until now, right? And what we could see the expression of these PSO2 receptors in the neurons which are processing the odor-related information. Now the question is, and why this PSO2 receptor expression is there? And if you think about how do we sense these olfactory signals, or how do rodents sense the signals? The rodents sniff at a high frequency rate, and we also modulate our breathing pattern to sense, to get a clear perception of the odors that we smell. And because of the modulation of that breathing pattern, it is possible that these receptors are also getting activated because of the mechanical changes or the sensation that may happen because of the changes in the breathing. And this might be contributing to the olfactory perception. And once we are able to establish this fact, I think we will have enough work for the next 10, 20 years. So this is exactly what we are focusing on right now, and that's why we are very excited about this year's Nobel Prize. And here are the references that are listed on the Nobel Prize website. And I'm sorry, I took 2 minutes more than they've given. I can answer the questions now. And thank you very much for attending the talk.
Looks like nobody has any more questions. Thank you so much, sir, for this wonderful talk. I sincerely hope audiences have taken valuable insight from this talk.
Okay, thank you very much for giving me this opportunity. Thank you. Thank you all.